Hi class, this is Dr. Lisa Carney Anderson, and we are continuing our discussions of the pulmonary system. And now that we've gotten the air into the lungs and diffused that oxygen into the blood, we're going to talk about how that oxygen is transported to the tissues, and then the CO2 is transported back to the pulmonary system. So here are our learning objectives. We want to talk about the partial pressure gradients that permit gas to diffuse between the alveoli and the tissue. I want you to be aware of the typical partial pressure values for alveolar, arterial, and venous partial pressures. Partial pressures being the way we discuss oxygen and CO2 levels. We use partial pressure kind of like a concentration. Then we have one of the most important diagrams in this chapter, figure 1322. I want you to be able to explain the relationship between alveolar ventilation and partial pressures. Then we're going to talk about hemoglobin and how hemoglobin is really responsible for the major content in your blood. You have dissolved content, which is in the form of PO2, but really the bulk of your content is bound to hemoglobin. So we want to compare and contrast some important terms. Saturation, which is a percentage of hemoglobin occupied by oxygen, and oxygen content, which is actually the amount of oxygen in mils. And then we want to finish up by talking about the transport of carbon dioxide. So I'm going to show you a whole bunch of graphs and try to make that more manageable. I'm going to ask you some questions, and basically those questions should help you see what to get out of the graph, and then we'll talk about the graph. So we want to talk about gradients that allow oxygen to diffuse from the alveoli to the blood. And then once we get the oxygen in the blood and take it down to the tissues, how do we go from the blood to the tissues? And then how is CO2 diffusing? Are there gradients for CO2? So this is a picture from your textbook, and I want you to notice we're going to start up at the top in the air. And we have some P sub O2, P sub CO2. Those are the partial pressures of oxygen, partial pressure of CO2. And to calculate a partial pressure, you take the total atmospheric pressure. So at sea level, that's 760 millimeters of mercury. In Minnesota, that's maybe 740 millimeters of mercury. And you multiply that times the fractional concentration. So there's about 20% of the air is oxygen. 20% of 760 is 160 millimeters of mercury. That's the partial pressure of oxygen. That's how much of the atmosphere is exerted by oxygen. Then notice PCO2, very, very tiny, less than 1. So we're not breathing in very much CO2. We're breathing out a fair amount of CO2. That implies that the CO2 we measure comes from our metabolism. So that ambient air comes into the body. First it gets humidified, just entering the airways, and then it goes down and mixes with the air already in the alveoli. Remember, there's always air that you can't blow out, and that sort of stabilizes the gradients in the alveoli. So now we have PO2 is 105 millimeters of mercury, PCO2 is 40 millimeters of mercury. That's the amount of the total pressure. That's what's being exerted by oxygen and CO2 respectively. So in the venous blood, it's actually in the pulmonary artery, but it's venous blood, PO2 is 40. So there's 105 millimeters of mercury oxygen in the alveoli, but 40 millimeters of mercury of oxygen in the venous blood. So there's an inward concentration gradient from the alveoli to the blood. And oxygen is going to diffuse into the body, into the blood. A lot of it's going to hitch a ride on hemoglobin, but the partial pressures are eventually going to become pretty equal. So notice PO2 in the pulmonary vein, it's arterial blood, but it's in a vein, is up to 100 millimeters of mercury. Likewise, venous blood has a PCO2 of 46 millimeters of mercury. In the alveoli, it's 40. So there's a concentration gradient out. CO2 diffuses out. And O2 and CO2 have different diffusion constants. So PCO2 only requires a small gradient in order to diffuse out. So notice the oxygen gradient is much. Okay. Once again, I do not have a sneeze button. The oxygen gradient is much bigger. However, oxygen doesn't have the diffusion constant that CO2 does. So really the two diffuse in about the same time. So PO2 of the arterial blood is about 100. PCO2 is about 40. So you notice there's a difference between the alveoli and the arterial blood. Because there's a little bit of mismatch in ventilation and perfusion, and there's some normal shunt and that kind of thing, typically there's a 5 millimeter of mercury difference between what's in the alveoli and what's in the pulmonary vein. So if the alveoli are 100, then the pulmonary vein is 95. If the alveoli are 102, then the pulmonary vein is 97. That's a normal difference that we see in everybody. We don't see that difference in CO2 because there's some CO2 is able to diffuse quite well. And that's kind of complicated. So I'll just leave it there. So the arterial blood goes back to the left heart, is pumped out to the systemic arteries, and then comes in contact with the tissue capillaries. So the oxygen level in the cells is low, so oxygen diffuses out of the blood into the cells to support metabolism. Eventually that oxygen has to go into the mitochondria, has to be the final electron acceptor in oxidative phosphorylation. We need a certain level in the mitochondria so that aerobic metabolism can take place. And with a breakdown of glucose and other carbon substrates, we produce CO2. The cells produce that CO2. The CO2 then diffuses out into the blood, and then venous blood becomes 46 millimeters of mercury. So we use up the oxygen and the PO2 drops. We produce CO2 and add that to the capillary blood, and so then venous PCO2 goes up. Goes through the right heart, goes through the pulmonary arteries, goes back to the alveoli, and we get exchange again. So we have diffusion gradients between the alveoli in the blood and the tissues in the blood. And those gradients support delivery of oxygen to the cells and the return of CO2 back to the lungs. Now, when we have blood floating through a capillary, how long does it take the blood to get loaded up with oxygen? So we see that in this diagram here. My x-axis is the percent of the capillary length. So I'm 20% through the capillary, 40% through the capillary, 60% through the capillary. When I'm all through the capillary, I'm 100% through the capillary. And notice we start at a PO2 of 40, because that's the venous PO2 of blood. And I eventually get my blood up to 100 millimeters of mercury, which is about what the alveolar PO2 is. Notice that that happens when I'm about a third of the way through the capillary. I have plenty of reserve. So even if I start exercising and the blood starts going by faster, I have plenty of time to use that diffusion gradient and load up the blood with oxygen. However, that's in a healthy lung. If I have a diseased lung, let's say I have swelling, let's say I've lost some tissue, let's say I have inflammation, let's say I've damaged the alveoli so that the alveoli are thick. All of that is going to interfere with diffusion. And so it's possible that I could get all the way through the capillary and my blood would not be fully loaded up with oxygen. So what could I do to help this person? Maybe I could increase the diffusion gradient. I could give this person some supplemental oxygen, then their alveolar PO2 would be higher than 100, and then maybe I could overcome the diffusion impairment this person has because of their lung disease. That doesn't work for everybody, but for some people with lung disease, you notice they will carry around supplemental oxygen. You're essentially trying to increase their diffusion gradient. Alrighty, now what in the world is alveolar ventilation? And when alveolar ventilation changes, how do partial pressures change? Well, just as cardiac output is the amount of blood you pump per time, ventilation is the amount of air you breathe per time. We calculate minute ventilation by taking a tidal volume, which is a normal breath, times respiratory rate, and we get V dot E. When we put a dot on top of the V, we're making it a ventilation. Ventilations are per time. Now, if I want to know exactly the air going down in the alveoli and participating in gas exchange, I take a tidal volume and I subtract off something called dead space. Dead space is the part of the tidal volume that's staying in the airways and the mouth and nose, and it's not participating in gas exchange. So if your tidal volume is 500 mils, your dead space is about 150 mils. I subtract that off, multiply times the respiratory rate, and then I get alveolar ventilation. So if my tidal volume is 500 and I'm breathing 10 times a minute, then my minute ventilation is 5,000 mils per minute. If my dead space is 150, then tidal volume of 500 minus dead space of 150 leaves 350 mils actually going down into the alveoli. If I'm breathing 10 times a minute, then my minute ventilation is 3,500. So 3.5 to 4 liters a minute is a typical alveolar ventilation. So in this diagram, that's what we see. In the middle of the x-axis, you see alveolar ventilation of 4. That's a typical alveolar ventilation. If my alveolar ventilation is higher than 4, then I'm hyperventilating. Now, you may hear the word hyperventilate and think about people that are kind of breathing fast and shallowly, like they're panting. That's actually not hyperventilation. When people hyperventilate, they're increasing their rate and their depth of breathing so that they increase their alveolar ventilation. And look what happens when you hyperventilate. You load up a little more on oxygen, so partial pressure of oxygen goes up, and you blow off CO2, so the partial pressure of CO2 goes down. Now, why should that be a problem? Because our plasma pH is related to the amount of CO2. And if we don't have enough CO2, then our pH is too high, and we have a condition called alkalosis. Now let's go back to our 4 liter a minute alveolar ventilation. Resting CO2 40, resting O2 100 millimeters of mercury. What happens if we don't breathe enough? What happens if our alveolar ventilation decreases down to 1? Well, notice that when we don't breathe enough, then we don't bring in enough oxygen, and our partial pressure of oxygen drops. Not only that, but our partial pressure of CO2 rises because we're not getting rid of the CO2. What's the problem? Well, when you add CO2 to the blood, it decreases the pH. It makes it more acidic. And so then we have so not only are we hypoxic, but we have an additional problem called acidosis. So the point I would really like students to see is that breathing is not only important for getting in oxygen, breathing is important for getting rid of CO2. And getting rid of CO2 is just as important as getting oxygen in, because CO2 determines your plasma pH levels. And proteins are very susceptible to the pH environment around them. Now, once we get this oxygen in the blood, how are we carrying it? 2 ways. Dissolved, which is a very small part, and bound to hemoglobin. And hemoglobin is the major part, so hemoglobin is more important. If we didn't have hemoglobin and just had to rely on dissolved oxygen, our cardiac output would have to be 67 liters per minute. Now, a resting cardiac output is 5. An elite athlete may be able to get their cardiac output up to 25 liters a minute, maybe 30. But there's no way any human being can have a cardiac output of 67 liters per minute. So we cannot live without hemoglobin. Here's a picture of hemoglobin. Hemoglobin is a gigantic protein with a heme group. And the heme group is a sort of a pigment with an iron group in it. And that's what allows hemoglobin to bind oxygen. And one hemoglobin molecule can bind 4 molecules of oxygen. And it does so with a property we call cooperativity. And how do we know that? Cooperativity means once one oxygen molecule binds, then it's easier for the other oxygen molecules to bind. Well, the reason why we know that is because of the shape of this curve. This curve is a sigmoid-shaped curve, and sigmoid-shaped curves imply cooperativity. This is how it works. My x-axis is PO2. And you see sort of a venous PO2 of 40, an arterial PO2 of 100. And then my y-axis is saturation. What's the percentage of my hemoglobin binding sites that are occupied with oxygen? Well, if our arterial PO2 is 100, then our saturation is about 100%. Now, it's hard for people to be exactly 100% saturated, but there may be 99% saturated, 98% saturated. Pretty darn close. And you notice the PO2 goes to 80, and we're still in the high 90s. It goes to 60s, and we're still at over 90% saturated. We get to be about 50, maybe we're 85% saturated. At 40, we're 75% saturated. So we've dropped from 100 to 40, we've dropped 60 millimeters of mercury, and saturation has maybe dropped 15 percentage points. Now go from 40 to 20. 40 to 20, saturation drops from 75 to 25. So it's dropped 50 percentage points. So this hemoglobin molecule has two very different behaviors. When oxygen is high, hemoglobin is a binding molecule. And the more oxygen binds, the easier it is for more oxygen to bind. Below a PO2 of 40 or 60 or 50, hemoglobin transitions to an unloading molecule. It's a releasing molecule. And the more oxygen is released, the easier it is to release even more oxygen. So this sigmoid-shaped curve, this S-shaped curve, is very typical of molecules that demonstrate cooperativity. This gives hemoglobin a dual nature. In the lungs, where the oxygen is high, hemoglobin is a binding molecule. In the systemic capillaries, where the oxygen is lower, hemoglobin is a releasing molecule, and it's just gorgeous. So here's a little more about dissolved versus bound. So you breathe in the oxygen, this oxygen diffuses across the membrane. It enters the plasma and becomes the dissolved form. And it's the dissolved form of oxygen that exerts PO2. Now that oxygen diffuses into the cell and finds a hemoglobin binding site. Now oxygen becomes part of the hemoglobin molecule and it no longer exerts PO2. So that means another oxygen molecule can diffuse in and take its place. So oxygen continues to diffuse in, and when all the hemoglobin sites are bound, then oxygen diffuses until the concentration gradient is equalized. And then no more oxygen can come in until we get new blood coming in that doesn't have oxygen. Then at the tissues, hemoglobin has to release the oxygen, it has to dissolve, and now exerts PO2. It dissolves into the, diffuses into the plasma, diffuses out of the capillary, diffuses into a cell, diffuses into the mitochondria, and when the mitochondria uses it up, then it no longer exerts PO2, so more oxygen can leave the blood and come in. So there's a total content of oxygen. Most of that content is bound to hemoglobin. Some of that content is dissolved. So dissolved plus bound equals total content. However, it's the dissolved oxygen that actually exerts the property we call PO2. Once oxygen is bound into hemoglobin, it's no longer oxygen, it's oxyhemoglobin. And then it no longer exerts PO2. So I'm going to tell you about a couple of people. One person's name is Mary, and the other person's name is Frank. And Mary has 12 grams per cent of hemoglobin. What does that mean? That means every deciliter, or every 100 mils of her blood contains 12 grams of hemoglobin. That's the way we express the concentration of a hemoglobin, that then we don't have to deal with the fact that Mary may be a smaller person and may have a smaller blood volume than Frank does. So Mary has 12 grams per cent of hemoglobin, and Frank has 13 grams per cent. So Frank has more carrying sites for oxygen. So at a PO2 of 100 millimeters of mercury, I'm asking you, Mary or Frank, who's more saturated? At a PO2 of 100, who has a higher content, Mary or Frank? Well, if Frank has more hemoglobin, he has more binding sites. But saturation is a percentage. So at a PO2 of 100, Mary is nearly 100% saturated, and Frank is nearly 100% saturated. Their saturations are actually the same. However, because Frank has more hemoglobin, he has more binding sites for oxygen, and at the same PO2, he has a higher content. And specifically, he has a higher bound content. Actually, their dissolved content should be the same. And why is that? Because they have the same PO2. So saturation is determined by PO2. Content is determined by PO2 and hemoglobin concentration. What about at a PO2 of 60? As well, Mary will be less saturated, but she'll have the same saturation as Frank. And maybe 85% saturated, something like that. And Frank will only be 80% saturated, but he has more hemoglobin, so his content will still be higher, and specifically the bound content. So once again, saturation is determined by PO2. Once you're 100% saturated, you're 100% saturated. However, content is determined by both saturation and PO2 and hemoglobin concentration. So it's important to be aware of this for example, when people have anemia. Somebody might have anemia, but you put a device on their finger called a pulse oximeter, and it says they're 99% saturated. You might say, well, this person is fine. However, until you take a blood sample and see what their hemoglobin concentration is, you actually don't know how much oxygen they're carrying around. So 7.6 is something that I want us to do in class and really discuss and work on. And it gives you a bunch of scenarios, and I ask you about partial pressures, that's what I mean by blood gases, saturation and content. So if you think PO2 and PCO2 are changing, you have to ask yourself, is this person breathing any different? If you think the saturation is changing, you have to ask, well, did something happen to hemoglobin or did something happen to PO2? All right. Let's talk a little bit about carbon dioxide transport. Now, some of your carbon dioxide dissolves, and some of it binds to hemoglobin, though it binds to a different place than oxygen binds to. But the majority of your carbon dioxide is converted to a molecule called bicarbonate, and this is how it happens. CO2 and water, with the help of an enzyme called carbonic anhydrase, can come together and form a molecule called carbonic acid. H2CO3 is carbonic acid. It is a weak acid and dissociates into a hydrogen ion and a bicarbonate ion. So as CO2 is converted into bicarbonate, a hydrogen ion is made. And this is why when we retain CO2 and CO2 builds up, it causes acidosis. And this is why when we don't have enough CO2, we don't have enough hydrogen ion, and we have a situation called alkalosis.